Welcome to Simplified UPSC by PPP, where we listen, learn, and grow together. Detailed lecture notes in English and Hindi, plus practice questions for prelims and mains, only on our Telegram channel. Link in description. Like and subscribe for more.
Welcome back to the Deep Dive. This is where we take essential source material, find the absolute core of the knowledge, and give you that shortcut to, well, to really being informed. Now, before we get into today's material, which is exceptionally dense, it's a foundational look at NCERT biology for competitive exams, I do have a really important and humble request.
Yeah.
If you find what we do here useful, please just take a second to like this deep dive and share a comment. It helps a lot. And if you're able, please think about subscribing and joining our paid membership.
It really is the support that lets us keep creating this kind of, super useful content for you.
It is. And on that note, we have to give a huge, sincere shout out to all of our existing members.
Absolutely. Your support is the bedrock. Thank you, truly.
So today's mission, we are going laser focused. It's a rapid but really rigorous dive into Class 11 biology, and we're hunting for those specific facts, the definitions, the little linkages that pop up in general science questions.
Right. Think of this as a synthesis. We're connecting the dots from taxonomy through cell biology all the way to plant physiology.
And picking out those tricky little distinctions.
Exactly. The ones that separate, just knowing the topic from knowing it well enough to ace an exam.
Okay, let's dive in. The biggest question of all to start, what is life? You see all these properties, growth, metabolism, reproduction, but what's the one characteristic that truly defines a life form?
So the one defining feature, the one that's in every single living thing and completely absent in every non-living thing, is cellular organization.
Cellular organization.
That's the non-negotiable threshold.
So why isn't it something like growth or reproduction?
Well, because they have exceptions. Growth, for instance, can happen in non-living things. Think about a mountain accumulating dust or a crystal growing.
Right.
And reproduction doesn't work because, sterile organisms exist. A mule, for example, or an infertile human, they're definitely alive. But if you're talking about life, you are always talking about cells.
Got it. So that defines life in general. But the sources point out a really fascinating distinction when you get specifically to human beings.
Yes. And that distinction is self-consciousness.
Okay, so not just consciousness.
No, all organisms are conscious of their surroundings. They sense light, chemicals, what have you, but a human being is uniquely aware of themselves, of their own existence. That introspective awareness is the defining property of our species.
That's a huge leap from the physical to the, well, almost philosophical.
Yeah.
Now, let's talk about classification, taxonomy. Give us the quick rundown on taxa and the really crucial rule about the hierarchy.
A taxa is just a grouping, any valid category. Dogs is a taxa, mammals is a taxa, wheat is a taxa.
Okay.
Now, here's the critical part that can feel a bit counterintuitive. As you move up the hierarchy, so from species to kingdom, the number of common characteristics actually decreases.
Wait, hang on, that feels backwards. Shouldn't a huge kingdom like Animalia have more defining traits?
It's about breadth, not similarity. Think of it like this. To be in the species Homo sapiens, you share almost every single trait with another human.
Makes sense.
But to be in the kingdom Animalia, all you need to have in common with, say, a jellyfish and an earthworm is that you're multicellular, eukaryotic, and you eat other things. That's a massive drop in shared traits. The broader the category, the fewer things everyone has to have in common.
Okay, that clicks. Higher up means more diverse. Now let's hit a direct fact that comes up in exams. Biofertilizers.
Right. You need to know the difference between Agaricus and Nostoc. Agaricus is just a mushroom. It's a fungus. It doesn't fix nitrogen.
What Nostoc?
Nostoc is a cyanobacterium, a blue-green alga. And it's incredibly important because it has these specialized cells called heterocysts.
And they do what exactly?
They can take nitrogen straight out of the atmosphere, which is totally inert, and convert it into compounds that plants can actually use. That is the core function of a biofertilizer.
And to round out this first section, a little biology, geography crossover. What's the defining feature of an equatorial forest according to the source material?
It's the structure, the presence of tall, very closely set trees, and their crowns form a continuous canopy.
A roof, essentially.
Exactly. A dense, closed roof that completely controls the ecosystem below it. It defines everything.
All right, let's move from the forest to inside the cell. Prokaryotes versus eukaryotes. What are the key details that separate these two designs?
Prokaryotes, so your bacteria, blue-green algae, they're basically defined by what they don't have.
It's
A well-defined nucleus. Their genetic material is it's naked, it's floating in the cytoplasm. They also often have these little extra circles of DNA called plasmids, and their ribosomes are the smaller 7ds type.
And eukaryotes are everything else. Protists, plants, us.
Right, and we have a proper membrane-bound nucleus protecting our DNA. And our ribosomes are the larger 80s type. That nucleus is the real dividing line.
Okay, let's build on that. Biomolecules, specifically proteins. How should we think about them structurally?
Proteins are heteropolymers. That just means they're long chains built from different units, in this case, amino acids, linked by peptide bonds.
And this structure has different levels.
4 levels. Primary is just the sequence. Secondary is simple folding. Tertiary is the really crucial 3D functional shape. And quaternary is when multiple chains team up.
Okay, quick fire facts. Protein abundance. 2, you absolutely have to know.
This is a classic exam question. Collagen is the most abundant protein in the animal world. It's the scaffolding for skin, bone, everything.
But not for the whole planet.
No. If you look at the entire biosphere, the winner, by a long shot, is rubisco.
Rubisco.
Yeah, ribulose, bisphosphate, carboxylase, oxygenase. Plants need absolutely massive amounts of it because it's the central enzyme in photosynthesis that captures carbon, and frankly, it's not very efficient at its job.
So they just make more of it.
Yeah.
Let's talk energy, metabolism. What's the simple way to split anabolic and catabolic pathways?
Think of it as construction versus demolition.
I like that.
Anabolic pathways are construction. You're building complex things like proteins from simple parts. And building always consumes energy.
And catabolic.
That's demolition. You're breaking down complex things like glucose into simpler parts. And demolition releases energy, which the cell captures in ATP.
ATP, the universal energy currency. And what accelerates all this? Enzymes. How do they work their magic?
So enzymes, which are almost always proteins, are catalysts. They don't change the final result of a reaction. They just make it happen, well, millions of times faster by lowering the activation energy, the energy needed to get the reaction started. They do this by grabbing the substrate at a special spot called the active site, forming a temporary enzyme substrate complex, and that makes the whole process much, much easier.
Okay, let's move into the plant world. Plant physiology. Water transport in roots can seem a bit abstract. Let's make it clear.
Right, you have 2 main pathways, Apoplast and Symplast.
Apoplast first.
The Apoplast pathway is kind of the easy street. Water just flows through the non-living cell walls, never crossing a membrane. It's a continuous system until it hits the Casparian strips in the endodermis. Think of these as a watertight gasket. They force the water to finally enter a living cell, to cross a membrane, and that's when it enters the Symplast pathway.
Which is water moving through the cell.
Exactly. Cell to cell.
Okay, another critical distinction for exams. Transport direction. Xylem for water, phloem for food. What's the difference?
This is a big one. Xylem transport is always, and I mean always, unidirectional, only up, from the roots to the rest of the plant. But phloem transport, now that is beautifully complex. It's bidirectional.
You go up or down.
Up or down. It all depends on the source-sink relationship. A source, like a leaf, makes sugar. A sink, like a growing fruit or a root, needs it. The phloem is like a smart delivery service, sending food wherever the demand is highest.
This next topic is where it gets really interesting for me. Biological nitrogen fixation. So prokaryotes convert atmospheric nitrogen, N2, into ammonia using the enzyme nitrogenase. What's the catch?
The catch is oxygen. The nitrogenase enzyme is unbelievably sensitive to oxygen. It gets destroyed by it, basically.
So this whole critical process has to happen in an anaerobic environment.
It absolutely has to.
Then how on earth do bacteria in a legume root nodule, which is sitting in oxygen-rich soil, pull this off?
They have a secret weapon. A molecule called Leghaemoglobin.
Leghaemoglobin.
It's an oxygen scavenger. It's a pigment very similar to our own hemoglobin that floods the nodule and binds up any free oxygen. It essentially creates an internal oxygen-free bubble to protect that vital enzyme. It's incredible biological engineering.
That detail feels like a guaranteed test question. Just for completeness, can you list a few free-living nitrogen fixers?
Sure. You have aerobic ones like Azotobacter, anaerobic ones like Rhodospirillum, and of course the cyanobacteria we mentioned like Anabaena and Nostoc.
Okay, before we zoom out, let's quickly nail down the difference between C3 and C4 photosynthesis.
Right. C3 is the standard path. First product is a 3-carbon compound, PGA. The whole thing happens in the mesophyll cells with rubisco doing the work.
And C4.
C4 plants are specialists. They first fix CO2 into a 4-carbon compound in the mesophyll using a better enzyme called pepase. Then they shuttle that compound into special bundle sheath cells.
And that's where the Calvin cycle happens.
That's where it happens. It's a way of concentrating CO2 right next to rubisco and protecting it from oxygen. It's a clever adaptation, usually for hot, dry climates.
So C4 is basically a two-step process to boost efficiency.
That spatial separation is the C4 signature move. Precisely.
Now let's pull the lens back. The sources for these deep dives often touch on general science, those crossovers you need for exams. Let's connect some dots, starting with physics. The Higgs boson.
Right. Its discovery was so important because it helps us understand the fundamental question of why elementary particles have mass.
It fills a gap in the model.
A huge gap. Without it, the standard model of physics was incomplete. The Higgs is what gives mass to other particles.
Okay, geography. Where do we find the world's best fishing grounds?
They're found where warm and cold oceanic currents meet, not atmospheric oceanic.
And why there?
Because that mixing churns up nutrient-rich deep water. That causes huge plankton blooms, which feeds the whole food chain, which means tons of fish.
Back to biology. A practical question. Why do fruits last so much longer in a cold storage unit?
It's a simple biological reason. The cold temperature dramatically slows down the rate of cellular respiration.
Which is the process of decay, basically.
Exactly. Respiration breaks down the sugars. If you slow that internal process down, you extend the fruit's shelf life.
Okay, one final one. Perception. Which phenomena in the sky are actually optical illusions?
The classic examples are the apparent size of the sun at dusk.
It looks huge on the horizon.
But it isn't. Also, the color of the sun at dawn and the twinkle of stars. All of them are just distortions caused by light refracting as it passes through our atmosphere.
Amazing. Okay, what a session. We've covered so much ground: the definition of life, the dominance of rubisco, the crucial difference in xylem and phloem transport.
And that amazing Leghaemoglobin oxygen mask.
I love that.
It just shows how everything in biology is connected. Protein chemistry, plant physiology, it all fits together. That synthesis is really the key.
So before we sign off, here's one final thought for you to consider. We've talked about movement, like in a cell or a muscle. Now think about the difference between just movement and locomotion.
It's a powerful distinction. All locomotion is movement, but not all movement is locomotion.
Explain that.
Well, think about it. Your heart beats, that's movement. Food moves through your gut, that's movement. But you aren't changing your location. Locomotion is when the entire organism moves from point A to point B.
It's a reminder that even the simplest terms have layers of complexity.
Exactly. Think of all the different ways to achieve locomotion. A snail's slime, the way your muscles work, all different drivers, but the same outcome.
A fascinating concept to reflect on. And that is our deep dive. If you enjoyed this, if you found it useful for your preparation, please, you know what to do. Hit that like button and share a valuable comment.
And we'll gently say it again, if you are able, please do subscribe and join the paid membership. It helps us keep this resource going for everyone.
It truly does. And one last time, to our current members, thank you. We'll see you on the next deep dive.
Thanks for watching. Follow us on Telegram and Instagram. Don't forget to like, share, and subscribe.